system. They don't actually come from the pancreas, they come from the brush border of the intestinal cells, which is the little microvilli and the simple columnar of your duodenum. So what happens is amylase comes out and can yield a bunch of disaccharides, but to really get down to your monosaccharides, we still have to wait on a couple other enzymes from the brush border. Okay, but anyway, what our focus is today is just how do we get to maltose. Why am I telling you about these other enzymes is because you want to realize we still need to go farther down to glucose. And all kinds of starches have some kind of glucose and sometimes some other types of sugars. But don't forget, you have the pancreas working to release maltase, sucrase, and lactase, or excuse me, maltose, sucrose, and lactose, but not until these enzymes can we break the disaccharides into monos. And we need them in monos to be able to absorb, okay? So that's just general information to remember and know. But again, what our experiment today is focusing on this amylase under 3 conditions, okay? So, remember how an enzyme works, called a catalyst. Okay, so enzyme today is amylase. Okay, and we're like looking at this question today in 3 situations. We're asking this, okay, starch, which we're going to call the substrate. Okay, this needs to be broken down or hydrolyzed. Okay, remember, all digestion, you have to get hydrolysis going out of for proteins, carbs, and your fats. Okay, you're going to always be breaking things down and then absorbing them or using them right away. And it depends on the state of the body. That's metabolism. Okay, but anyway, starch is a substrate in this case, and the product we're looking for is called maltose. Okay, maltose, that's a sugar. So this is a disaccharide that we can call the product here. Okay, so here is a disaccharide. Okay, and this we would call what? Polysaccharide. Okay, so our question today is involving 3 situations changing the situation of amylase. 1, we're going to look at a reaction of starch, and we always are water. You know, water is the base of the entire body. So water's everywhere. So you think we're going to have starch and water and no enzyme, and we're only going to have an 11-minute test. We're going to ask the question here for our hypothesis, do we think without an enzyme would starch hydrolyze to maltose? Then we're going to look at another question, room temperature, the presence of amylase. Well, that probably would help to get substrate to product. And then finally, if boiling the enzyme to very high temperature, boiling it with high temperature for like 5 minutes, which is a big deal. Okay, we're going to see if the reaction still works after we've boiled the enzyme. Okay, so backups for background information. Again, enzyme is a very specialized globular protein that are all over the body. We need enzymes for like everything, to produce energy, for absorption, for our sodium potassium pump. We need enzymes all over the place. Okay, and how do they work? They can be used over and over. Now, they're not going to last forever. We do need to replenish enzymes by resynthesizing those proteins, but this enzyme can be used over and over. So what you want to think of in this case, the substrate is starch and the enzyme is amylase. So the enzyme amylase is a globular, very specialized protein having a very specialized binding site for starch, right? And so it binds to the product and it's going to allow those internal reactions to occur. And in this case, it's hydrolysis, adding water to break bonds. And then the products, we say, are maltose. Okay, now this is showing us just generalized model. You got substrates and products, and you get just very generalized here. So what you want to understand right in your introduction are how do enzymes generally work. They have an active site, right? And that's going to be very specific for their substrate. And then how do they work is the fact that they're going to lower the amount of energy costing that cell. All of these reactions that we think about and talk about all over physiology require energy. So with the energy of activation is just saying, what's the energy requirements to move this reaction? So let's say starch to maltose without an enzyme, let's say the activation energy is 10 ATP. Well, what an enzyme like amylase could do is say, hey, I can do this reaction for 2 ATP. You see, it's lowered the amount of energy. So that's how I explain how enzymes work. Is without this enzyme here, let's say starch to maltose would take 10 ATP. That's a lot of energy. Okay? But what an enzyme does is say, I can help you do this for 2. And I don't know the real numbers. It's just the general concept. That we're going to lower that energy activation. So we're going to lower how much it costs for that reaction to move forward. Okay, that's how enzymes work. Okay, so here is showing us where you've got the rate of enzyme activity across temperature. Okay, because what are we going to do today is boil it. So who knows what's going to happen if we boil a globular protein such as amylase for 5 minutes? It's going to stop working. Now, why is that? There's a special term, what we call proteins that unravel, denatured. Okay, so what we're looking at is, are we going to denature amylase in that third trial? When we boil amylase, what we're really thinking here is, are we going to actually denature it and will it not work? So if an enzyme doesn't work, do you think a reaction would proceed within 11 minutes? Maybe not. It's almost like not having one at all. Okay, so just trying to get you to start thinking here. So the physiology is not hard. Okay, it's not hard. Just are we going to denature the enzyme or not? You know, and the point is, yes, we need an enzyme. Okay, but what the hard part is running this experiment and analyzing the results. Okay? So, another thing that I want to show you is when temperatures are too low, it's not necessarily that they're denatured, but they just become inactive. They're just not able to maintain those active sites as well. But when I say denaturation, that's a true unraveling. So you see, like, this could be a globular protein functional enzyme that is folded in a very special way to fit its very specific substrate. But what happens is if we change the temperature, because if you think about it, these proteins are synthesized under normal conditions, right? Normal body pH, normal temperature. So if we start changing what they're used to, their bonds are not going to stay formed. And so what they can do is unravel. So a functional globular protein is no longer functional if you've disturbed its three-dimensional shape. So we call that denaturation. Now, one other little point. What do you think reversible denaturation is compared to irreversible?
Irreversible could be fixed back?
That reversible can be fixed back. Is that what you said?
Yeah.
Yeah. Reversible means we can reverse it back if the temperature or pH were not too extreme, so mild changes in temperature and pH, and they get restored, we might see reversible denaturation, meaning as soon as we restore the temperature and pH, the enzyme folds back. It works again. But in situations where it's an extreme pH, extreme temperatures, like boiling is pretty extreme, we're going to probably cause what we call irreversible denaturation. It's not going to fold back. It is just destroyed, right? Does that make sense? Okay, that's just the idea that you can ask, are we talking about reversible denaturation? Can it go back to functional ability? Or irreversible? Not real. Okay? Irreversible. Alright, so that's not hard, really. Okay, so what we're going to do is this. This is your experiment purpose. Okay, so here's your purpose is, you want in your background for this paper, you want to think. And those that didn't turn in your glucose hepatic portal system paper today, you're going to have to write a report on this one. Okay, so you want to make sure in your introduction I would talk about why do we eat starch? It's because we need glucose. Why do we need glucose energy production? And why do we need enzymes? Because all these reactions would never occur in time if we didn't have them. Okay, and you talk about maybe some amylase and how it comes from saliva, but also pancreas. Okay, and that we need enzymes. And then the information about how enzymes can be denatured if under certain conditions. Okay? So, all of that's your background. Our purpose today is to then ask 3 situations. The absence of amylase. Do we think starch will get hydrolyzed to maltose? Okay, number 2, do we think it will in the presence of amylase? Okay, and number 3, in the presence of boiled amylase. 100 degrees Celsius is like, what, 400 or 500 degrees Fahrenheit? Or more, I don't know. 600, what's the boiling point? I can't remember. So anyway, starch solution will produce maltose, possibly. So if we think more than one of these solutions will produce maltose, which one do you think would be fastest relative reactions? Okay, so how are we going to do this is what gets tricky. But do you understand the basic concept? We're trying to see, 1, do we really need an enzyme? And 2, what if we boil the enzyme? Is it going to cause denaturation? That's really what we're kind of looking at. Okay, so come up with some hypotheses for 1, 2, and 3. How are we going to do this is what's very tricky. Okay, so before I even tell you how we're going to run the experiment, let me show you. Well, this is our big question today. Is starch going to be broken down to maltose in 3 situations? That's our question. These are all clear solutions. So how the heck are we going to know if starch disappears because it's breaking down and if maltose appears because it's being released? And they're clear solutions. How are we going to figure this out with any special chemical testing? Well, we're going to use reagents. Okay, so the way we're going to do this is use a reagent that we know detects starch. We're going to find out the color change. And we know there's another reagent that can detect maltose, and it's Benedict's. Okay, so what we're going to first do is learn how to establish controls of the experiment. Okay, positive controls, meaning these are the variables we're looking for to interpret our results. Okay, so let me show you what I mean. What we're going to be doing is using both Lugol's and then Benedict's to really ask the same question. So the first 11 minutes, what we're going to do is use Lugol's with these spot plates. Okay. Well, we got to know what Lugol's does and how it reacts, and how it should only change color if starch is there. Okay, so you're going to do this to learn how Lugol's detects starch. This is called identifying your control. It's the very first part of your procedure. Okay, so control ID for starch. Okay, what you want to keep thinking about is this. We have starch, and we're trying to get it broken down to maltose. We know that we're changing the variable of the enzyme, okay? But what the other components here are H2O. Okay? So what we're going to do is using Lugol's and Benedict's in each of these solutions, starch, water, and maltose, to determine how these reagents tell tell us who's there. Okay, so let's see what I mean. Starch in the beginning of our experiment should be there. We're going to ask if starch breaks down to maltose. So what you're going to learn today is you're going to use these spot plates for the actual experiment, but the very first part is called control identification. You're just going to use these 3 little wells, they're shallow wells, and you're going to put H2O, starch, and maltose. You notice there's nothing to do with enzyme amylase at this point. Okay? We're only looking at how do we identify these pieces of our question, because they're clear solutions. How are we going to know if starch is broken down and maltose appears? Okay. So what you're going to realize is Lugol's or IKI starts as a light yellow. What I'm going to tell you is that it should react with starch to produce a positive color control. That's a dark, brilliant, bluish black color, like a inky black. So what you're going to see is this. You have identified your positive control for starch. Do you see? Because and this isn't the experiment, this is just so we can run the experiment. We're going to have to figure out how we're going to know starch there and when is maltose going to appear. And you'll see what I mean as we talk about the experiment. Okay, so this is the first part. You have to say, how do I know in this whole experiment today, where is starch in every situation? Well, if black is there and Lugol's is there, that means starch is there. Okay? We're also going to say a control identification, positive starch, also for the product. Because then we want to see, is this product going to be released? So we're going to use Benedict's. Okay, and you remember you got to boil Benedict's. So you have to take test tubes, not just your spot plate, and you're going to take 3 tubes, see? And again, this is not your experiment. Okay, we're going to put H2O in one, starch in the other one, and maltose, in another one. And we're going to add Benedict's. Remember, it starts kind of that light blue color. But what we're going to see here, which one do you think turns like a reddish orange?
Maltose.
The maltose. Okay? We haven't run an experiment. All we are doing is learning how to use our reagents to detect these variables here. Okay? Now, does this make sense? We got to know what to look for. And this is going to make sense. The first half of the experiment, we're going to use Lugol's. Then we're going to confirm by using Benedict's. Okay. So this is Lugol's.
Sorry.
And this is Benedict's. Okay, remember you got to boil these. Now, once you have done all of this, then you're ready for your experiment. Okay? Then you get another 3 test tubes. Now what we're going to do is add and your handout walks you through this. I'll show it to you. I've kind of summarized it in my pre-lab here, but the details of each procedural step is in your handout. But basically, this is what you're going to do. You're going to 3 tubes, A, B, C. Absence of amylase, so we're just going to have water, right? Presence of amylase, well, that should be some water and amylase. And then the presence of boiled amylase is water and amylase that's also been boiled for 5 minutes. And then you let it cool. Okay. So now we're ready to run our experiment. But again, we're not going to add starch just yet. We're going to actually create a timed race. And so we can't add starch to our actual test tubes until we're absolutely ready to go. Okay, because what you're going to do, you're supposed to only do 1 test solution at a time. You can do that, but if you get organized, I can help you show how you work together and it goes faster. Okay? But look at this. This is how you're going to do it. Whether you do them separately, test 1, test 2, test 3, meaning water, amylase, boiled amylase, separately, you can do that, or you can run this together if you're set up and organized. So let me show you what I mean. First thing you'll want to do is get your hot plates going, because these are spot plates here, okay? So what you're first going to do, somebody's already labeled them. I like lining them up like this, okay? Because what we're going to do is test for 11 minutes. You'll want to label, absence of amylase, presence of amylase, and then the boiled amylase. You see there's 3 tests that we're going to run a race. And from time 0 to time 11 minutes. Okay? So what you could do, you could run them one at a time, but you could, after you've done your control IDs, then you set up your actual test tubes. You know what you can do if you have 4 students. 1 student per tube, and then another student can time you. Because what you're going to do is this. As soon as you're ready, you're going to add starch to our test solutions, right? That's time 0. So what each of the 3 students that are going to be in control of, let's say you're in charge of just water, you're in charge of just room temperature amylase, and you're in charge of boiled amylase. Okay, so once you have your solutions and you're all ready, you've labeled this, you're going to time. So right away at time 0, you're going to add some starch, and all the details of how much and how to do it is in your procedures. You're going to add a little starch to these solutions, right? Right away. You're going to shake it and then drop it right away. Wait a minute, and you're going to be timing, and then at minute 1, each student drops in their respective well. Of course, you're going to water, you're going to amylase, and you're going to boiled amylase. You see? Then another minute goes by. You see the reaction is going on in the tube. But what we're going to have, I forgot to tell you, we're going to first put Lugols in every single well. Then we're going to run this test. So if you think about it, when we add Lugols to all of these wells, right, then we're going to add the starch, shake it up time 0, put it in there. What color should it immediately turn? The black inky. So we know we're starting with starch. The point is, every minute we go, we're trying to see if starch breaks down, right? So minute 1, we drop. Minute 2, we drop again. Each respective tubes. Minute 3, minute 4. What we're looking for is if the black disappears. Because what does that indicate?
The breakdown of starch.
Yes, the starch is getting hydrolyzed. So our first way that we're going to answer our question today is using Lugols in a timed manner, drop by minute by minute by minute. They all start black because they all have starch right away. But we're going to see if any of them disappear and go back to the original yellow color. Then the main second part of this experiment is taking all 3 of those tubes, right, that have been reacting with starch, and add equal amounts of Benedicts, boil them. Which one produces maltose is our question there. Well, think about it. Do you think, let's say test tube water stays black. Yours stays black until about minute 3 or 4, your amylase. And yours stays black the whole time. Well, which one of those races then should then end up with maltose at the end? Which one of those components? If there's only one that the black was beginning to fade away, what we just said is that indicates starch is broken down. So what should there then be if starch is broken down?
Maltose.
Maltose. So the other part of the experiment is to confirm. You're answering the same question, but just a different way. You're going to confirm, well, the one that disappeared, that should be the one that turns bright orangey red in Benedict's.
So when we boil it, then
Yeah.
say that, confirm that the sugar is in there.
Right, right. So that's what you're doing. Okay, so don't get this so confused. I know this is tricky. The first thing you are supposed to do is do what I've already shown you what you're going to see, but go ahead and just do it. Okay, your control identification of starch and that of maltose.
And that'll be in the 0 well?
Yeah, just any 3 wells that you want, and then you can rinse them out and dry it and just start again, okay? Does that make sense? I'll help you understand if you still don't.
I don't know how we're supposed to drop them all at the same minute.
Today, can we on down
That's
here?
like impossible. Like we're gonna be standing here like,
Move on, it's fine, just don't
We just
think-
have to prepare that one night, but we're gonna. I don't know.
Okay, so do you
It's
see
really
that
hard.
this is part of your procedure, but it's not the actual experiment. So what we're calling this is
thank you.
control identification of starch, and we're going to call this control identification of maltose. And I should say positive. Positive control identification for the other part of our variable in our
I can
experiment.
upload
You
this.
see,
So
is
you
your
can
maltose.
listen.
Okay, which is that. This is your positive. And this, of course, is your positive. So that's why you have 6 tubes. You're going to have 3 to do your control and 3 for the actual experiment. Okay, so if you're writing this paper, what you've got to do is be able to explain how you did this. Okay, now if you go back, you can look at
I'm so glad I did.
the actual handout that this is going with. Is this. Okay, this is the detailed, procedure. What I've done is just try to do a synopsis and get you to understand that you can work together if you want to run them all at the same time. It's okay if you want to just go step by step and do one solution at a time.
It's hard to
Okay.
do it.
So you see
It's
this
hard
is
to
telling
do
me
it
how to
alone because you
identify
have to
starch,
drop it all
right?
together.
How to identify maltose. Okay, now you're ready to run the real experiment, right? So you're gonna begin to get all this stuff set up. And it's typically 2 mLs of like everything. All of the pipettes are graduated by milliliters.
Do you have the drawing mask? Meeting the drawing. You wanna see it? Yeah. Just for like.
Okay, let's do it. So the first thing is get your hot plate going.
I can do that later.
Get your hot plate going. Come up with hypotheses.
Yes. I know we don't have an iPad. Yes, we got. Yeah, bring that over here. I can post this one and continue later.